Welcome to our webinar on phase and connectivity analysis. My name is Bastian and I'm your presenter for today. I'm hosting this webinar together with Kidist as today's moderator and Laura as assistant moderator. In this webinar, I would like to present theoretical and practical aspects of connectivity and other phase-based methods that are common in EEG research. As you will learn later, we will look at common methods that quantify functional connectivity, and I will emphasize on the understanding of how it is computed. These methods comprise of some assumptions that are easily violated. Therefore, two of my main goals for this webinar are to raise awareness about the issues that are to be considered before and during analysis. But I would also like to provide guidelines to what can and cannot be learned from the results. In the hands-on session, we will create a connectivity workflow and apply it to real data. Along with this pipeline, we will learn how to use some of the modules in Analyzer that provides this methodology. Note here that parts of the modules that we will use are only available starting from Analyzer version 2.2.0. I will first introduce fundamental terms and concepts that we need to know about, but also more background knowledge that is important to understand the connectivity methods. I will then focus on the computation of synchrony that is the basis for many connectivity methods, and then we'll go through an example analysis that will show how the methods can be used to explore the connectivity, but also phase locking in our data. Finally, I will go through most important caveats that accompany this type of analysis and provide some practical solutions. After the presentation, we will have some time for questions. One of the most characteristic features of the brain is that it's highly interconnected. Large groups of locally connected neurons that are activated together, and those groups will tend to entrain into a synchronized rhythm. Such rhythms can occur at different frequencies and amplitudes and at different parts of the brain. Neurons of a local group are also connected to neurons that are more distant. These long-range connections are realized through axons and synapses that provide a rich anatomical structure. This kind of connectivity is called structural connectivity. However, the interaction between areas is not only determined by the anatomical preconditions, but can vary depending on the function they shall fulfill. For example, if a specific task requires frontal and posterior neural groups to interact, this interaction can be dynamically adjusted to meet these criterions. This type of connectivity we call functional connectivity. Such connections can be unidirectional or bidirectional, and one neural source can be connected to many other neural sources. In such a dynamic and complex system, it is generally not easy to understand how the different parts interact with each other and who causes what. Therefore, in EEG research, we mostly look at single pairs or even multiple pairs of neural groups, but we mostly ignore all the interactions that also might play a role. This already shows the limitation of functional connectivity that we need to keep in mind when interpreting our results. When we identify two brain signals to show connectivity, we never know whether they are also causally related, or whether they are only activated simultaneously through a third group. For most functional connectivity measures, we also do not know how they are directed. So whether one area influences the other, or whether it's the other way around. Generally, we look at how similar two signals evolve over time, and based on this judge whether or not they are connected. There's also another class of connectivity methods that try to overcome this limitation to some degree. They evaluate the directedness of connectivity by estimating how well one signal can predict the other, and vice versa. This class of connectivity is called effective connectivity. However, in this webinar, we will be only looking at undirected measures that fall in the class of functional connectivity. When we quantify functional connectivity based on EEG signals, so on the level of electrodes and not neural sources, we need to take some extra precautions. One major problem when computing connectivity on the sensor level is volume conduction. I will get back to this subject in more detail, but the general problem is that we often measure the signal of one oscillating source at many different sensors. Such spurious correlations interfere with our analysis and distort the picture of real connectivity that we want to investigate. We will learn about steps in our analysis, how to mitigate these effects and avoid making false interpretations. Another major problem is the problem of the common reference. All electrodes record signals always relative to a reference electrode. All electrical activity that is prominent at the reference electrode, including oscillations, will be present in all other electrodes. These correlations likewise are spurious and interfere with our analysis and the interpretation of our results. Later, we will also see what precautions to take to avoid these effects. Another fundamental aspect that is common to most functional connectivity methods is that they quantify synchrony. As mentioned before, local groups of neurons are active in a synchronized manner. Synchronized means that tens of thousands of neurons are entrained in the same rhythm. In the EEG signal, we observe such rhythmic activity as oscillations at different frequencies. And the raw EEG signal can be understood as the sum of many of these oscillations. Most functional connectivity measures quantify the interaction between EEG signals on the basis of these oscillations. That's why many methods are based on frequency domain data. Oscillations are assumed to interact if they are synchronized. So, for example, in the case of these traces, both start oscillating at a certain time point, and these are also highly synchronized. And this is because they have a constant phase difference. Their lead and lag relationship is stable over time. Synchronization thus is measured by quantifying how constant the phase differences between oscillations are. Because connectivity is often based on frequency domain data, we need to transfer our EEG signal into the frequency domain. If we have a stationary signal or can assume quasi-stationarity, so EEG signals that are made up of constant oscillations, such as this one, we can apply the FFT, or the Fourier transform, which calculates the average frequency content within a given time window. The resulting frequency spectrum can express the strength or amplitude with which each oscillation is present in the signal. For example, we see that this signal consists of an oscillation at 1, 8, and 30 Hz. However, oscillations in the EEG are often not stationary and change over time, as these transient signals. To get a better insight into the temporal evolution of the oscillations, we can use the wavelet transformation. It can estimate the spectral content for different frequencies, but also for every time point that is captured in the signal. For example, in this time-frequency map, amplitude or power is color-coded, and we can see how it changes over time. This time-resolved spectrum is in particular important for connectivity analysis because connectivity patterns are often not stable over time, and these patterns may not be visible when mixed with time intervals that do not show connectivity. Both of these transformations can result in complex coefficients or complex frequency spectra. For the FFT, these are called Fourier coefficients. For the wavelet transformation, these are called wavelet coefficients. They express the spectral content for each frequency, and in case of the wavelets, also for each time point. The coefficients are complex values that have a real and an imaginary part. In combination, they can represent not only the amplitude, as in these images, but also they can represent the phase information of the oscillation. So let's see what this means and how a complex value can represent both phase and amplitude at the same time. So let's take the sine as an example oscillation. It has a constant amplitude, let's say 1, and each point on the curve can be defined with respect to the origin, the latest positive zero crossing, which we have here. We call this the phase of the oscillation and can specify it in radians between 0 and 2 pi. One cycle is completed if all phases occurred once and a cycle contains a positive and a negative peak before it repeats. The time it needs to complete one cycle relates to the frequency of the oscillation, and this duration is also called the period of the oscillation. This oscillation can be represented as a vector on a circle. And the vector has a length that corresponds to the amplitude of the oscillation, and it points into a direction at an angle that corresponds to the phase at the origin of the oscillation. We denote amplitude here with A and phase angle with theta. We can uniquely describe this vector by using a coordinate system. And for that, we need at least two values, one for the y-coordinate and one for the x-coordinate. And here we make use of complex values that consists of two parts, a real and an imaginary part. So this type of value gives us the possibility to store two coordinates at once. One coordinate is stored as the magnitude on the imaginary axis, and the other coordinate is stored as the magnitude on the real axis, and both coordinates make up the real and imaginary part of the complex value here. Given the circular representation of the oscillation, we can uniquely describe every phase and amplitude by the combination of a sine wave and a cosine. The elevation on the imaginary axis, as described by the sine function, and simultaneously the variations on the real axis, as described by the cosine function, which is a sine function only shifted by 90 degrees, define in combination every point on the circle. Therefore, the sine on the imaginary axis and the cosine on the real axis can define every possible phase angle and represent the complete oscillation when scaled with the factor amplitude. You may find these formulations in a shorter notation in the literature using the Euler's formula. These vectors in complex space have to be defined for each frequency when the time domain data is converted into the frequency domain data. And that's exactly what our frequency decomposition tools such as the FFT and the wavelet transform are doing. And finally, the amplitude and the phase of this vector can be extracted using these formulas. So this was already some information, so let's summarize what we learned so far. In this session, we are concerned with the methods of functional connectivity. These quantify the degree of synchrony of two EEG electrodes or neural sources, where a higher degree of synchrony indicates high levels of connectivity. With these methods, we do not have the means to imply causality or directionality, and should be aware of this fact when drawing our scientific conclusions. Furthermore, it is important to consider the issues arising from volume conduction or the common reference, as these distort our connectivity results by inserting false correlations. Most measures are based on the frequency or time frequency domain data. Raw EEG signals in the time domain need therefore to be converted to either frequency or time frequency domain. This can be achieved with the wavelet transform or the Fourier transform. Frequency and time frequency domain data is represented in complex values, which can be understood as storing coordinates in complex space to represent phase and amplitude information simultaneously. So let's continue with the next section. In this, we will explore in more detail how synchronization is measured. Many connectivity measures are based on frequency domain data, but synchrony can also be based on the time domain data. Time domain signals, for example, are our raw or preprocessed EEG recordings, which can be converted into the frequency domain with, for example, the Fourier transform, the FFT. Whether or not these signals are synchronized can be assessed with these two quantities. In the time domain, this is the cross correlation. In the frequency domain, this is the cross spectrum or the cross spectral density. These two quantities actually contain the exact same information, but only exist in two different domains. Therefore, they're called Fourier pairs. Some measures of synchrony that are based on the cross spectrum or the cross correlation are subsequently being normalized. This is done to achieve comparability between channel pairs and to squeeze the values in a meaningful range. The level of synchronization can then be extracted from these normalized quantities. For example, you might want to extract the amplitude or the power spectrum of the normalized cross correlation, or you might want to compute the magnitude squared coherence from the normalized cross spectrum. At the heart of all this are these two processes. These are measuring the temporal correlation or synchronization. In my view, the most intuitive approach to understand how exactly synchrony is measured is to look at the cross correlation in the time domain. So let's have a look at it. In order to compute the cross correlation, we actually perform two steps. One is the time-lagged correlation, and the other is an average. Okay, so let's look at how the time-lagged correlation is computed. We only need to understand the basic principle of correlation here. So we have our time series x and y, and x and y are our EEG traces and consists of t values. For example, all our data points in one segment. The correlation simply multiplies all values in signal x with all values in signal y, only that the values in signal y are shifted in time by a certain amount, which we denote with the letter tau. And then we simply add up all the products. We denote this process by this star. And we might understand this as if we were shifting across over y and then summing all the products of the overlapping values. And this is maximal at total overlap. It is maximal here because all positive values in one signal correspond to all positive values in the other signal, which makes a positive product. And all negative values in one signal are matched with all negative values in the other signal, which also makes a positive product, summing up to the maximum here. Only if we shift the signal further, more negative values are matched with positive values and they make a negative product and the sum will decrease until it reaches a minimum at the antiphase, where all negative values are matched with all positive values. If all time lags are once taken into this function, we receive a central peak, such a typical cross-correlation function having a central peak and two recurring side lobes. Now let's look at two signals. Let's look at two signals that are actually different. So these are synchronized at a higher frequency, but signal X also has a prominent oscillation on a lower frequency. The oscillation at this lower frequency is not contained in Y, and therefore both signals cannot correlate at this frequency. In the resulting cross correlation, only the low frequency is not visible, even though X is also part of the products, and only the oscillation that is synchronized is visible. Typically, we would compute the cross correlation between two channels in one segment, and do this for all available segments. In order to receive the cross correlation with the beta signal to noise ratio, the average is computed. If the temporal structure is consistent across many trials, so if the peaks of the cross correlation occur with the same temporal lag between signals for many trials, this will also show up in the average, indicating synchrony between the signals. The distance between the peaks, if consistent, also informs about the period of the oscillation at which synchrony is measured. And with the period, we can also infer the respective frequency. Let's now look to the frequency domain and see how the cross spectrum is computed. The cross spectrum is calculated also in two steps. One is the computation of the complex difference vector, and the other is the vector summation. In order to compute the difference vector, we need the complex frequency spectra of both signals. So we need here either the complex Fourier spectrum, which can be computed from the FFT, or the time-resolved complex wavelet coefficients given by the wavelet transformation. The difference vector is computed via the multiplication of the complex spectrum with the conjugate of the complex spectrum of the other signal. But let me visualize what that means. So we have our two complex frequency spectra consisting of a real and imaginary part. And as we have learned, we can represent a sample value at a given time and frequency with these complex vectors. They point into a certain direction that corresponds to the phase of the signal and have a certain length that corresponds to the amplitude of the signal. Now, to compute the difference vector, the phase difference, which is also called the relative phase, is computed. This is simply the subtraction of the two phases and yield an angle that lies between the two vectors. Note that this difference corresponds directly to the time lag between two oscillations. The next step is to compute the amplitude of the difference vector. This is simply multiplying the length of one vector with the other. And finally, both are combined to the complex difference vector that has a relative phase and a length that corresponds to the product of the two amplitudes. Typically, the cross spectrum is calculated as a sum across segments. Given frequency domain data, we have one complex vector per frequency and also per segment and per signal. The difference vector between the signals is then calculated as described here and then summed up. The summation in the complex domain can be understood as if concatenating the individual difference vectors from end to start. And the resulting vector will follow the path of the concatenation from the origin to the end. This yields a vector with a phase and amplitude that is specific to the combination of the individual difference vectors. If all phase lags between the signals are constant or similar across segments, then the difference vectors will point all in the same direction or similar directions. In this case, the magnitude or the length of the concatenated vector will be large, indicating high synchrony. If the oscillations are not synchronized, so phase differences are random, the individual difference vectors will point into random directions. And the magnitude of the summed vector here will be small and eventually approach zero, indicating a low degree of synchrony. The computation of the cross spectrum is the most important part of most synchrony measures and is crucial to the understanding of many connectivity methods. Let's now proceed to the next section that introduces normalization. Both the cross correlation and the cross spectrum are often normalized. The normalized version of the cross spectrum is the coherency, while cross-correlation is simply called normalized cross-correlation. The denominators used for normalization are values that are proportional to the amplitude of the individual oscillations that are involved. For the normalized cross-correlation, the denominator is the product of the root mean square of the two oscillations. And in case of the coherency, the two power spectra are used. Normalization has the advantage that the resulting scale is actually meaningful. Now, 0 means a total lack of dependency, and 1, or in the case of the normalized cross-correlation, also -1, means complete linear dependency, which only occurs if both thicknesses are identical. Normalization also achieves comparability. Now the values can be compared between channel pairs or conditions that have different power values. With this, I would like to finish the second theoretical section and turn to the practical part. In this part, we'll first turn to the magnitude squared coherence, and then look at the available connectivity transformations in Analyzer. For the hands-on session, we will use data from one subject that performed in a semantic priming paradigm. And the experimental paradigm consists of an auditory prime, a delay period of 1600 milliseconds, where only a fixation cross was shown, and a visual target. The subject was asked to respond to the target as fast as accurate as possible, indicating whether or not the object shown in the image is small enough to fit in a real card box. In some trials, the auditory prime would be semantically congruent to the target object, in others not. In such a task, we might expect that top-down cognitive control areas in the frontal lobe functionally connect to other task-relevant brain regions. For our example analysis, let's hypothesize that in anticipation of the task and the upcoming stimulus, such anterior-posterior networks might be established already in the time interval preceding the target. The dataset we will work on is already pre-processed, and our goal is to explore the connectivity structure in the time interval preceding the visual target. Since we are not interested in the primary priming effect, the N400, we can take all available trials that are completed correctly. Before we step into the data analysis, I would like to draw your attention to one more pre-processing step that will help for our connectivity analysis. I mentioned before that one major caveat is the problem of volume conduction. The conductive properties of the tissue between the neural source and our electrodes lead to the fact that multiple electrodes, or even all, pick up one and the same neural source. For connectivity, this is a particular problem since synchrony between sensors might reflect the same oscillation and not real neural interaction. Signals that are mixed in this process lead to dominant low spatial frequencies in the EEG data. And on the voltage map, they hide most of the local activity and only show one global voltage distribution. In order to minimize these effects, we can apply the current source density, also known as the surface Laplacian operator. It rejects part of the data that originates from deep or distant sources and estimates current flow at the dura. On the voltage map, this leads to highlighting of more local activity that has previously been buried. However, note that in particular neighboring electrodes will still suffer from effects from volume conduction, and we should take extra precautions when interpreting connectivity between closely spaced electrodes. Another helpful feature of the current source density is that after the transformation, the data will be reference-free. That means whatever the online reference or a subsequent new reference was, the impact of any previous reference will be cancelled out. The data is then reference-free. This can be understood by the fact that the CSD is an operation that is done across electrodes, and common influences that might originate from the reference are subtracted from each other and cancel out. In many cases, the CSD provides a good basis for connectivity analysis that is more robust against spurious correlations originating from volume conduction or the common reference. However, note that although we will base our analysis on the CSD data, this step might also have a downside. And whether or not the CSD is an appropriate pre-processing step depends on your data and research question. Sources that are distant or widely distributed are attenuated by the CSD. It might, however, be that these are the sources that are actually informative regarding your research question. Therefore, it might be a good idea to test your methods on the basis of the CSD and also on the raw or filtered EEG data. Okay, let's now go get into Analyzer and perform our analysis. So here I opened Analyzer in the version 2.2.0, and as introduced, I have here one data set from a semantic priming paradigm. The raw data was sampled to 256 Hz, visually inspected, and filtered in a passband between 1 and 30 Hz. We have already excluded ocular artifacts and pooled all correct and artifact-free segments in this node. You can see that the segments are being logged to the visual target. This is the respective marker. And the segments have a length of 3 seconds. Now, as a first step, I will apply the current source density to attenuate the impact of volume conduction in our data. You can find the transformation under Transformations, Data Preprocessing, CSD. These parameters here concern an interpolation step that is applied previously to the application of the CSD. It shall support a better estimate of the true surface voltage distribution. The spline parameters that we find here specify how the interpolation shall be performed. You can find more information on these parameters in our user manual and in the literature that is cited therein. You can also choose to keep channels that are not located on the head or do not have valid head coordinates. As I don't have such channels, I will now don't change anything and click OK. You can see that the traces are changing quite significantly, and also an ERP based on the CSD would look quite different than on EEG data. Now, based on the CSD, I will use a transformation that converts our time domain signal into the time frequency domain. And I will do this with the wavelet transformation, which can be found here in the frequency and component analysis group. Sorry, wavelets. I will use the continuous wavelet transform and choose the following set of parameters. I will stick to the complex Morlet wavelets, and the wavelet layer with the minimal frequency will also be at 1 Hz. The maximal frequency shall be at 20 Hz. So we'll be able to explore connectivity within this frequency between 1 and 20 Hz. In order to save some computation time, but still have some reasonable resolution across frequency layers, I will choose 15 frequency steps. I will not change any of the other parameters that we can, as the defaults should be fine for our example analysis. However, if you would like to know more about this dialog and the wavelet transform in general, you can watch the recording of the wavelet webinar hosted on our webinar channel. So let's continue. In this dialog, we are asked to choose the output of the wavelet transformation. As we have learned, the coherency is based on the complex coefficients. These contain an imaginary part and a real part, and in combination can store the phase and amplitude of the oscillation. Therefore, we choose first the option wavelet coefficients, complex values. At this point, I will not use any normalizations, as we need the raw wavelet coefficients for the coherency analysis. However, in special cases, you can use this panel to normalize and rescale your coefficients proportional to a reference interval. Otherwise, we've set all options and we can finish the transformation. Okay, let's quickly rename this node so we know what it contains. Now, although this data is complex in this node, Analyzer chooses to already show the extracted amplitudes of the complex values. This is a general principle. So whenever Analyzer is supposed to display complex data, it chooses to display amplitude, so the lengths of the vectors that we have been discussing. The reason for this is that not all quantities, imaginary part, the real part, amplitude, or phase information can be intuitively displayed simultaneously, and a decision has to be made what to display. Analyzer chooses to display amplitudes. So from this time frequency maps, we can already see some strong alpha oscillation that are present before the visual target. This is good to know, because only if we have sufficiently strong oscillations, we can hope to find synchrony between the channels. Okay, let's try and look in the first connectivity transformation in Analyzer. You can find the coherence transformation in the comparison and statistics group here. Starting from Analyzer version 2.2.0, this transformation will be available for frequency domain and time frequency domain data. However, it will not be available and grayed out here if the data is not complex, for example, when wavelet power was calculated. It will also be grayed out if there's only one segment available, for example, if an average was computed. So the first thing that we will ask in this transformation is to select the output method or the method that shall be applied. Let me switch back to the slides to explain what these are. All these methods are based on the cross spectrum that was introduced. The first method is the magnitude squared coherence. It is probably the most widely used method and is basically the squared amplitude of the coherency. So the squared length of the summed and normalized difference vector. It is always positive, as the length of the vector can never be negative and bounded by 1 due to the normalization. In reality, 0 is never reached, and 1 only if the two signals are identical. This value range poses some more challenges on the statistical evaluation of coherence values. But I will come back to this later. For the advanced user, it might be helpful to extract the magnitude of the cross spectrum or the phase of the cross spectrum, which can be the basis for other connectivity measures. The imaginary part of coherency is a common quantity to safeguard from effects of volume conduction. We will get back to this measure later and see how it can do so. Finally, you will have the option to choose the squared magnitude of the correlation coefficient, which is an alternative measure of the magnitude squared coherence. In the next dialog, we can choose the channel pairs we want to look at. Consider this full correlation matrix. The first row and first column consists of all available channels. And the pre-selections that you can find here refer to this matrix. The upper triangle and diagonal and lower triangle and diagonal select all available pairs. The difference between the two options is only the order of the channels in the channel pair. The order, however, is only important if we would have a directed measure of connectivity, which we don't. So at this point, it doesn't matter which of the two we choose. You can choose a seed-based like channel pairs selection by choosing the option row of or column of. This will select all possible pairs that include the selected channel in the drop-down menu. The difference between the options is again only the order of the channels. You can choose to select only pairs that span across hemispheres. Note, for that option to work, you will need to have valid electrode coordinates or labels corresponding to the 1020 system. And you'll have a manual selection option, which I will show next. In the manual selection, you can specify each channel pair manually. You'll have the option to make use of the pre-selections in order to modify the manual selection. For example, to disregard a subset of channel pairs. From this interface, you can also save your channel selection or to load one. The selections are saved in Brain Product's own Brain Vision graph file format, which are XML files, such as this one, and that basically store the channel labels that belong to a channel pair. Okay, so let's go back to Analyzer. As the first method in our analysis, I would like to compute the magnitude squared coherence. So I choose this option, and I prepared already a channel pair selection file, which I want to use. So I choose manual channel pairs and click Next. Sorry. As I have already prepared a file, I only need to load it via this load graph option. And all pairs that are specified in this file are all possible pairs that can be made with the frontal electrodes F3, F4, and FZ. Now the channel pairs are loaded in the list, and I have the opportunity to still change them if I want. But I don't. So I can finish this transformation. This will now compute for some time. The computation time of this transformation will depend not only on the amount of data, so the amount of sample in each segment, but also on the number of frequency layers that are in the wavelet node. But most significantly, it depends on the number of channel pairs that you have selected. In combination of these factors, this can quickly lead to an explosion in the computational requirements and exceed the memory that can be used by Analyzer. In this case, the transformation will prompt a warning and abort the operation. And then we would need to see how we can reduce the size of the data. For example, by choosing a lower sampling rate, shorter segments, fewer channel pairs, or less wavelet layers. However, here I have adjusted the parameters such that we don't run into this problem. Let's quickly rename this node to know what it contains. So we computed the magnitude squared coherence. The default color scale for paired metrics is Murray-Trarer, we can change the settings of this view by going to the context menu and setting of this view. We are in the time frequency domain, so we choose the time frequency tab and choose, for example, another color scale. I choose rainbow and it will interpolate the data. Now this is a personal preference of this color scale, and you can also stick to the default color scale or to any other. So I will apply this here and click OK. So as the coherence operation includes vector summation across segments, we only have one segment left here. So these are the time frequency map that we receive for each channel pair, and the channel pairs are indicated here in the upper corner. The level of the magnitude squared coherence is indicated by the colors, by the color scale. And you can see that in most channels, there is a strong coherence in the alpha band already. The color scale is set automatically for each view. So when I zoom into a channel pair, the color scale is rescaled and may be more appropriate for this view because it scales to the value range that we actually see. In particular, there's strong coherence in neighboring electrodes. For example, in the pair F3FZ, F4FZ, or in this one, F4FC2. Now, given what we learned already, I suspect that these high coherence values, but also generally the high and widespread alpha band coherence that we see in many channels might well be due to volume conduction. This is plausible because volume conduction effects might be most strong between neighboring channels and leads also to widely distributed coherence. These effects are partially, but not completely discarded by the current source density, which we've applied before. In order to attenuate further the effects of volume conduction, we can apply the imaginary part of coherency. Let me switch back to the slides to explain how that is done. So for orientation, we now already calculated the magnitude squared coherence, the first frequency domain measure that is based on the normalized cross spectrum, the coherency, and we'll now look at another measure that is based on the coherency, the imaginary part of coherency, a quantity that is less sensitive to volume conduction effects. The rationale why the imaginary part of coherency is less sensitive to volume conduction effects is the following. If two or more electrodes are recording essentially the same oscillation by means of volume conduction, the resulting oscillations at the sensor level shall be close to identical. That means that their phase differences are zero. In other words, synchrony occurs with zero phase lag. If oscillations were to be synchronized through the interaction of neural groups, we would expect that they will not have a close to zero delay, because the speed of interaction is limited by the conduction delays of the axons and synapses connecting both neural populations. In this case, we would assume that there is a non-zero phase lag. What does that mean for the imaginary part of coherency? Let's again look at the computation of the cross spectrum, which is the basis of the coherency. So we have again phase and amplitude information of two oscillations represented as these vectors, and the resulting cross spectrum represented as this difference vector. If the influence of volume conduction is strong, there's no time lag between the oscillations and their phases will be identical. In this case, the difference vector has a zero phase angle. However, if the oscillations originate from independent neural sources, they will have some lag and the phase difference will not be zero. Now, if we again look at the representation of this vector in complex space, we can observe that the difference vector that is recorded under volume conduction lies on the real axis here, and the imaginary value corresponding to this vector is zero. The imaginary part of this means that the imaginary part of the cross spectrum is also zero. And its normalized version of the cross spectrum, so the coherency, the imaginary part of that coherency will also be zero. So in conclusion, the imaginary part of coherency will be less sensitive to coherence induced by volume conduction because the corresponding value will be small or close to zero. However, again note that there are some secondary effects of volume conduction, and these may not confirm with this assumption that spurious synchrony only occurs at zero phase lag. These effects will still prevail when using the imaginary part of coherency. Note also that the imaginary part of coherency can be negative. So when a vector is pointing downwards, the imaginary value corresponding to it will be negative. Let's look at the effect in the analyzer. So essentially we need to re-raise the coherence transformation and choose another output option. We can do this by going to the context menu of the coherence node and choose Edit Parameters Copy. Here I only change the method to imaginary part of coherency and leave the rest unchanged. When this is finished, I expect to see that the imaginary part of coherency is less pronounced at neighboring channels as the magnitude squared coherence values were. If so, this indicates that the values of the magnitude squared coherence underlie the influence of volume conduction. Looking at the imaginary part of coherency is thus a useful control analysis indicating relations in the data that should be omitted when interpreting the data. So let's wait until this is finished. Okay, so let's quickly rename this node, indicating imaginary part of coherency. And I will also change again to the other color scale and interpolate the data. Okay, so let's look at a neighboring electrode pair. For example, F4, Fz. And also let's look at this pair in the magnitude squared coherence. So I activate magnitude squared coherence again, and then choose also F4, Fz. And here we see already a large difference. Let's close some windows and choose another tiling. So now we see that much of the connectivity that is displayed here in the magnitude squared coherence before the visual target is gone and not visible in the imaginary part of coherence. So here the values are close to zero. Furthermore, if we select a certain time frequency point in this map, we can also see in the connectivity graphs here in the lower corner that the strong alpha coherence is only visible between neighboring electrodes here in the frontal part. This, because they are neighboring, is also a good indicator that this is due to volume conduction. When switching to the imaginary part of coherence, we can see also that these connections are now visible across the whole head and not only locally strong. So that we can now see those connections could be explained because the large coherence values that come from volume conduction effects are now strongly attenuated and other real connectivity values are uncovered. So let me change the view. However, we see also, that there still is strong alpha coherence in the imaginary part of coherency. So we're looking at the imaginary part of coherency at many channels. So for example, the pair FZ-PZ still shows quite prominent alpha band activity or coherency before the visual target. And this pair is not neighboring, so it's distributed across the whole head. And in this case, I would now assume that this coherency that is shown here is not due to spurious correlation, so not due to volume conduction, for example. Now, there's one aspect of the coherency, the imaginary part of it, and also the squared magnitude that should be understood when looking at these images. And that is that the computation of it is influenced by the amplitudes of the underlying oscillations. Let's go back to the slides to understand why this may be a problem. So let's consider these 4 segments of artificial data. Within the segments, amplitudes are constant, but between segments, amplitudes vary. Both oscillations are synchronized, but their phase difference varies slightly. In this case, the lengths of the difference vector pointing upwards are larger than the lengths of the difference vectors that are pointing downwards. If we now look at the vector summation process, larger difference vectors, in particular here the first one, exerts the strongest influence on the direction of the summed vector, while the other vectors have a much smaller influence. Thus, the path that connects the origin to the end of the last vector is biased towards the direction of the longest constituent vectors, and thus to the segments or pieces of data that have the highest amplitudes. This does not only influence the angle of the vector summation, but also influences the length, and in consequence, the magnitude of the cross spectrum. And because this magnitude is biased, also the magnitude of the coherency is biased, even though it is later normalized to the power of the constituent signals. This dependency is in particular problematic when the research question requires to quantify phase synchrony, but also if you compare conditions where the overall power of the involved oscillations varies. In this case, the contribution of the connectivity that is amplitude related or related to the phase differences are not clear, and a more strict phase synchronization method is needed. In order to meet these difficulties, measures have evolved that ignore the amplitude. So, as you can see here, all individual amplitudes are of the same size, and also their difference, the cross spectrum, the difference vectors are also of the same size. So all constituent vectors have the same influence on the resulting vector summation. In this case, the cross spectrum only quantifies whether or not the phase differences between signals are stable across segments. A measure that it does exactly this is the phase locking value. It is not based on the raw wavelet coefficients, but on the wavelet phase. The wavelet phase is also a complex coefficient, but the amplitudes are all 1. I use a shorter notation here that you might also find in the literature. The difference vector from these normalized quantities also have the length of 1, and the resulting summation will not be biased by the amplitudes. The wavelet phase is extracted by dividing the raw wavelet coefficients by their own amplitude, resulting in an amplitude of 1. The phase locking is then extracted from the amplitude of the average vector. So here we have the average operation. Note that the final normalization step here is not needed, as all amplitudes that went into this vector average are already normalized. Again, for orientation, the phase locking value is not based on the coherency as the other two measures were, but it's directly derived from the cross spectrum. However, as we have seen just now, the complex time frequency domain data, in our case the wavelet coefficients, were already normalized. So this normalization step is skipped. In Analyzer, we do not have a dedicated transformation to compute the phase locking value, but we can follow a small pipeline with only 3 processing steps to arrive at the PLV, the phase locking value. The first step is to perform the wavelet transformation, along with the options to choose wavelet phase. This output is already the normalized complex vector. So for each time point and frequency, and for each segment, we have complex wavelet coefficients that all have an amplitude of 1. In the next step, we will use the correlation measures transformation that you can find in the same function group as the coherence transformation. It has a very similar dialog, and as the coherence transformation, only the output options differ. With this transformation, we want to compute the phase differences of both signals on the one hand, and on the other to compute the vector average. Both is achieved in one go with this transformation if we choose the normalized correlation. The normalized version of the correlation is unit-free and will therefore display no units, which is appropriate for the phase locking value. In the last step, we need to extract the length of the vector average, and that is done with the rectify transformation. Here we only need to select the available channel pairs. Rectification of complex values is identical to taking the absolute value of the complex vector or its amplitude. And for the PLV, this results in the length of the complex vector average of the relative phases. So let's apply this pipeline in the Analyzer. At first, we need to raise the wavelet transformation again. For that, I go back to the CSD node and raise the wavelet transformation. I will again choose the continuous wavelet transformation and will keep all the settings that we had before. And now choose the option wavelet phase, complex values. Note here that you can also choose to output the phase angles in radians or in degree. However, for our next steps, we need the complex values. So I stick to the wavelet phase option and click Finish. So don't be surprised by this display. It again only shows the amplitude of the complex data in this node. And since the color scale is set automatically, it goes from 1 to 1. And here all the values are only 1. So let's quickly rename this node to indicate its content. And based on this node, we can now raise the correlation measures transformation that you can find also in the comparison and statistics group below coherence. As explained in the slides, I will choose the normalized correlation and go again for the manual channel pairs. I again load my channel pair selection file and finish the transformation. When this is finished, I would expect that for some electrodes, the phase locking value draws a clearer picture as, for example, the imaginary part of coherency does. I expect this because the influence of the amplitudes can somewhat blur the synchrony values, not only across time, but also across frequency layers. Okay, so this takes a bit longer. And So bear with me until this is finished. Okay. So in the last step, we need to extract the amplitude of these average vectors with a rectified transformation. And you can find this transformation under data preprocessing and rectify. Here I already selected all channel pairs that I have, and click OK. I will also rename this node to indicate the phase locking value. Now again, I would like to switch to the Brainrow color scale. I do that quickly. And change the view a little. So here again, we can see that there's a lot of alpha synchrony in many channels. And we can also see that in particular for neighboring channels, again, there are high synchrony values. And this is because the phase locking value, like the magnitude squared coherence, is vulnerable to the volume conduction. For comparison, let me go back to the imaginary part of coherence, coherency, and choose just a second. And choose this pair here, F4, FZ. Oh no, F4, ZZ, this one. Just a second, I need to rearrange the view a little so I can show what I want to show. So this is imaginary part of coherence and the phase locking value, and we choose the same channel. So in the same channel pair, the F4 ZZ, we have a pretty noisy picture in the imaginary part of coherence. We have blobs of high synchrony, and in particular a high synchrony blob after the visual onset of the onset of the visual target. And this is also during the period where we would assume an evoked potential. So if I compare this to the phase locking value, we on the one hand see that the synchrony values that we have here are much more defined in one time frequency band. And furthermore, the blob of high synchrony after the visual target disappears. So this hints to the possibility that the high synchrony values in the imaginary part of the coherency is partly due to the amplitude modulation that we would expect from an evoked response here, and thus may not reflect actual phase synchrony between the oscillations as it disappears the phase locking value. Okay, so with this, I covered also the last connectivity method that I wanted to introduce. And let me go back to the slides for one more intermediate summary. So we learned about the current source density and that it attenuates the effect of volume conduction and also produces reference-free data. Both effects help to reduce spurious synchrony from the data. The CSD is a good first step in the connectivity analysis, but it should also be used with caution, as it can attenuate synchrony between sources that are of interest. We learned how the cross spectrum is computed and have seen how it compares to the time domain cross correlation. The cross spectrum is at the heart of many connectivity measures and is essential to understand the methods. We learned that we can use the imaginary part of coherency as useful analysis to safeguard our analysis from volume conduction effects. and also got to know a more strict phase synchrony measure that is independent of the amplitudes of the involved signals. Depending on your research questions, data, and study design, this property is important, and in some cases, it can paint a clearer picture of the underlying connectivity structure. Before I come to the last section of this webinar, I would like to mention one more measure. It is not a connectivity measure, but also based on phase. And we can derive it from the wavelet phase. It quantifies the locking of phases across segments and not synchrony between channels. This distinction is important because, although it measures something entirely different, it is often confused with the phase locking value. I'm talking about the phase locking factor. And the motivation behind this measure is nicely exemplified with the phenomenon of alpha blocking. So it is a common finding that when a subject focuses on the center of the screen and after some time a visual stimulus is presented, the ongoing alpha oscillations reset to the onset of that stimulus. If this happens in each segment, the phases on the ongoing alpha activity are tightly locked to that stimulus. And at the same time, alpha power reduces significantly. When averaging in the time domain across segments, only phases that are tightly locked to the stimulus will add up. This is the evoked response. Oscillations that are not tightly locked to the stimulus so oscillations that are induced, they will vanish when averaged across segments in the time domain. However, the amplitudes of the evoked oscillation depend on the amplitudes of the single trials. This is important to consider when testing phase locking to an event, because the portion of the evoked response that is only due to the phase locking is not easily distinguishable from the portion that is based on the amplitudes on the single trials. And comparing between conditions with differing amplitudes can be ambiguous. In order to find how strong phases are locked to the event, we need to look at how reliable the same phase occurs at a given time in each segment, or whether phases are jittered and are more randomly distributed across segments. This can be done with the phase locking factor. Let's see how it's computed. Consider an evoked oscillatory response and an induced oscillatory response. In the evoked response, the evoked response is not only locked in time, but also the phases are tightly locked, as indicated by the vectors again that all point into the same direction. The phases of the induced response are not. Similar to the phase locking value, here the aim is to ignore all the amplitudes and quantify phase locking only. For that, the individual amplitudes are normalized. This is also the same procedure as we have used for the phase locking value and results in the amplitudes that are all 1. Now, in contrast to the phase locking value, we do not calculate the phase differences between channels, so we do not quantify the synchrony or connectivity, but we directly average across segments within each channel. Therefore, phases that are locked to the stimulus, if the same phases occur at the same time across segments or in many trials, these phases will add up to a long vector average. If the phases are randomly distributed, as in the case of the induced response, the resulting vector of the concatenation will be small. indicating a low level of phase locking. So let's see whether the phases are also locked in our paradigm. So we don't have a dedicated alpha blocking experiment here, but I could speculate that at the onset of the visual target, or even at the preceding auditory prime, so somewhere here, phases are reset or locked to the onset of these stimuli. To explore this possibility, let's activate the wavelet phase node. Remember, this already contains the normalized wavelet coefficients. So the only remaining step is to compute, computing the phase locking factor, is to compute the average and then to extract the length of the average vector. Let's apply the average. I will choose here the entire set of segments. Although this again is only a simple average computation, this will take some time. And remember, this process is first of all done in the complex domain. And then we have also many wavelet layers that need to be averaged. So we again wait until this is finished. And now, as a last step, we need to extract the length of these vector averages using the rectifier transformation. Again, this can be found under data preprocessing rectify. Here I choose all available channels and click OK. I will rename this node again to indicate the phase locking factor. And here we can see that at the occurrence of the auditory prime and after the visual stimulus, phases are highly locked. So, for example, in frontal regions, as you can see here, in the alpha range, phases are locked to the onset of the auditory prime, that is at minus 1600 milliseconds. And in the occipital regions, phases are locked to the visual stimulus. For example, here in the theta range, or more centrally in the beta range. Okay. So with this, I would like to finish the hands-on session and continue in the slides. Before having a final summary, I would like to revisit the most important caveats that we encounter in a connectivity analysis. The problem of volume conduction and the common reference. Volume conduction is a problem for connectivity because we may observe synchrony between channels, here in blue, that reflect the same oscillation emitted by a single source, which is picked up then by both senders. Similarly, if the common reference picks up a prominent oscillation, this oscillation will be mixed into all other channels, and then this oscillation may emerge as synchrony between those channels. Both are major problems in connectivity analysis on the sensor level. The consequences of them, as well as approaches to avoid spurious connectivity, are subject to a large body of research. Some solutions to these problems are: to tackle the issue of the volume conduction, we can use the imaginary part of coherency, as we have done, on the one hand, and on the other hand, you can use also the current source density, which we also have done. However, whether or not they are used simultaneously is up to the research question. Another common approach is to use source reconstructed data that is also less affected by volume conduction. It also always helps to take the connectivity pattern that we can observe across channels into account. For example, if we find high correlation values between neighboring channels, we should take extra precautions. Because the problem of volume conduction affects all data at all times and sensors, we can contrast the data to a task irrelevant or neutral condition. In that case, the effect of volume conduction should be present in the task relevant and the neutral condition. In the difference of both conditions, the shared influence by the volume conduction should cancel out. The solution to the common reference problem is mainly to get rid of the common reference. This could be either reference-free data, as provided by the CSD bipolar derivations, or source level data, or to choose a reference scheme where channels in a pair are separately referenced. For example, using left and right mastoids respectively. Many connectivity measures, such as the coherency and the phase locking value, are based on the computation of vector averages. These vector averages, again, are biased by the number of segments or samples that go into the computation. This bias has been shown to lead to an overestimation of the connectivity estimate, in particular for small sample sizes. Furthermore, the length of the vector average can never be zero. This has strong implications for the distribution, which is not normal. This poses problems for parametric statistical tests and the estimations of significance. So one solution to meet these pitfalls is to choose an analysis design that levels them out. For example, to choose the same sample size for different conditions that are compared so that the bias is identical, or to perform statistics on difference waves between conditions so as to change the distribution of values. Non-normal distributions can also be Z-transformed to conform with the normality assumptions of many tests. Or non-parametric statistical tests can be used. These do not have specific assumptions on the underlying distribution and are thus not dependent on normality. Okay, we are at the end of the webinar, and I would like to summarize the more general aspects that I hope that could be learned from it. Functional connectivity evaluates synchrony between signals by calculating the phase differences between oscillations. On the sensor level, in particular, these results are vulnerable to physical interferences such as the crosstalk between electrodes, or to methodological limitations such as the dependency to amplitude variations. In order to avoid making false interpretations, we can use a multitude of analysis methods and also pre-processing steps that help to show the full picture of connectivity. And with this, I would like to finish my presentation, and we now have some time for open questions. Okay, so here we have one. So you mentioned that coherency can be based on FFT data. Can you show how to do this in Analyzer? That's a very good question, and yes, I can show you that. So the coherence transformation can, as I said before, also be based on frequency domain data, and frequency domain data can be extracted via the FFT, the fast Fourier transform. However, one prerequisite for the FFT is to choose data that is stationary. So in our case, for example, we could look in a let's look at the imaginary part of coherence. We could look in time interval where we expect that there is some constant oscillation. Let's say from -100 to in this interval preceding the visual stimulus. In order to get this interval, we would use a new segmentation step to extract this time interval. So in this case, I'm choosing the same settings that I've had before, and only change the time interval. For example, I'm choosing now 500 milliseconds preceding the stimulus. And this data can now be converted with the FFT to the frequency domain. And here the important parameters are for connectivity to choose only voltage and to produce the complex output. And we would not recommend to choose any of the other settings here, as we want the raw complex Fourier coefficients from this transformation. So we get here again the display of the extracted amplitudes of the complex spectrum. And based on this, we can again raise the coherence transformation. And yeah, we can use also the imaginary part of coherence. And now, because we have much less data than for the wave transformation, we can also choose a higher number of channel pairs. Let's take all of them. And yeah, this selection can be used. So if you tick this option, you will be directly shown the channel pairs view or the band channel pairs view. But I skip this step now and finish the transformation. So this is pretty fast. And here we get the spectrum of the imaginary part of coherence, which can be positive, which can also be negative. Now, for example, we can also look, if we mark this data, and we can use a transient view, for example, the band channels pairs view, and then we get the same display that we had in the time frequency map in the lower corner, the connectivity graph view with the scalp map with all the electrodes that we have and edges connecting those electrodes, and the color again indicates the value of the alpha band, which I've marked here, of the imaginary part of coherency. Now, to make this picture a bit more clear, we can also change the setting of it again by going to the context menu and change the manual scaling essentially. So for clarity of this picture, I will only look at the positive part of the imaginary part of coherence and apply this. Click OK. And now we have also handles here to the color scale. And now I can lower, for example, the limit of the upper limit to make the colors a bit more saturated, and use the lower level here and increase this level a bit so that coherency values that fall below this threshold become white. And white edges on a white background here disappear. So then with that, I can visually introduce a threshold on the connectivity value on the imaginary part of coherence to only show those connections that have at least some that cross this threshold. And here we see that there might be some frontal posterior network that shows higher connectivity values. Okay, so the steps for a coherence analysis based on the FFT is first take out the data that is quasi-stationary, then apply the fast Fourier transform, choosing the complex output with only the voltage option, and then you can apply the coherence transformation. So one question is, how can I export the maximal values in a specific frequency range? So this is a bit more complex. So I can show, first of all, I can show two types of export that we can do with our time frequency data. So I'm assuming that the question aims at these time frequency data, and for exporting that, you can always, for example, use the generic export option. So that would be via the export and then generic data. Then you can create a text file where you actually have all time and layer data in a text file, which you can also import to Excel, for example. However, what you can also do is to choose a specific time frequency range with a solution that we provide. This is the wavelet data export. And here you can, for example, here for the phase locking factor, but that's the same for other time frequency data as well. To raise this this solution, just a second. So we have it here, and here you can choose the different layers that you have. For example, you can choose an alpha band. Oh, that is a bit too large. And you can choose a time interval. Let's say we can do it the same interval as we had before. And now if you choose the point mean normalization, this export can, and you can also choose different channels that are available. This will then export the mean value in this time frequency space, and will be written to this text file, which you can then import to Excel or to another statistical software to perform your statistics on. Now, if you want to compute, if you want to extract the peak values in a given time frequency window, then I would suggest that you extract one wavelet layer. For example, you can do this if you are again on the time frequency, in the time frequency domain. You can use the transformation wavelet layer wavelet extraction, and here you can choose one of the layers that we computed. Let's take layer 12, and then now you still have frequency data in the sense that this only resembles the one wavelet layer, so one frequency, but also time resolved. And then you can specify peaks in this time domain and export these also via the peak information export that we have. Okay, I think we may have time for one more question. So can I calculate the magnitude squared coherence on average data? And this, well, this is not possible. And the reason is simply, if we look back at the magnitude squared coherence where we based our data upon, we have a number of segments, and we need also multiple segments to calculate the magnitude squared coherence. So in this case, this is only the complex wavelet data that we have, and as we've learned, this vector summation that is computed has to include multiple values. So it cannot be that you only have one segment and no vector summation or average can be computed. And therefore, it's the coherence transformation that we find here will also be grayed out if you only have one segment. So the answer to that would be no, you cannot. Okay, so with this, I would like to also close the questions section. And I have here a number of references and citations to literature that you can read. And they, I think, give a good overview of the methods of connectivity that we talked about here. And I also recommend to consult our analyzer user manual if you have more, if you want more detailed information on the transformations that we used. Okay, so with this, I would like to thank you for your attention, and you will soon receive a follow-up email with a link to the PDF slides and a video recording of the webinar. You can also watch this and past webinars on our webinar channel. And please do contact us at support@brainproducts.com if you have any more questions to this webinar or to other topics in Analyzer. So thanks again for watching this webinar, and I hope you enjoyed it, and that we'll see you around in the next webinar.